All right, so as soon as I get my stopwatch out, Vivek, obviously, is going to start here. Right, Vivek, it's all yours.
Good afternoon, everyone. My research title is Strategies to Transition to Organic Grain, Soil Health, Crop Production, and Profitability. How do I advance? Oh, sorry. So, grain farms are required to be managed using the standard organic practices for 3 years prior to certification, and that period is termed as organic transition. The certified organic grains fetch higher or premium prices, and the practices are considered to be more eco-friendly. However, the organic transition also comes with challenges. Farmers face with challenges such as fertility management, weed management, maintaining the yield, profit management, and so on. The typical organic farmers use tillage for weed control and making seed beds, and use animal manures and legumes for soil fertility. The case of Maryland, the conventional Maryland grain farmers majority of them use no-till and strict nutrient management plans and use cover crops, including legumes. So the eco-friendly starting point at Maryland begs the question of whether transitioning to the organic farming practices from the conventional practices would cause an increase or decrease in soil health, crop productivity, and profitability. So to find an answer to the question, we identified 4 different strategies that are along the continuum of different levels of soil disturbance and cover crops, in which the first treatment 1, uses traditional full tillage and basic cover crops. The treatment 2 uses reduced tillage and moderate cover crops. Treatment 3 uses minimum tillage and high biomass cover crops. And treatment 4 avoids tillage at all, and it uses alfalfa grass hay, perennial alfalfa grass hay. So these systems are being compared for soil health, crop productivity, and profitability. And with the study, we intend to find the optimum strategy, or which I like to call the sweet spot, that can make the organic transition profitable, practicable, and eco-friendly for the farmers. Thank you.
Let me know.
Go for it.
Ready to rock and roll. All right. So the title of this research is Energy Production and Waste Treatment from Food Waste and Black Water through Integrating Anaerobic Digestion, Hydrodynamic Cavitation, Microbial Electrolysis Cells, and Electrocoagulation. My name is Kirk Mahoney, and the co-authors of this research are Dr. Amar Hassani, my advisor, Dr. Stephanie Lansing, Dr. Naresh Kumar, and Dr. Greg Lorraine. So the buildup of biomass residue can cause substantial environmental burden without the appropriate treatment pathways. So to mitigate these concerns, research activities have focused on creating renewable, alternative, and carbon neutral energy sources that can treat the waste properly while having little to no detrimental effect on the environment. So our research focuses on a novel approach to combining electrocoagulation, hydrodynamic cavitation with gravity settling, anaerobic digestion with a microbial electrolysis cell to create renewable energy and for water reuse in a low tier capacity from a food waste and black water waste stream. So the first objective of this research is to 1, test the ability of hydrodynamic cavitation to increase the organic material availability from a food waste and black water waste stream. Our second objective is to determine the effect of a combined ADMEC to enhance the energy production from food waste and black water after being treated with hydrodynamic cavitation. Our third objective is to assess the ability of electrocoagulation to improve the water quality. So what we found was that with the ADMEC and the HDC was that when we applied settling and cavitation to the food waste and black water waste stream, we actually saw a large increase in the energy production in the form of methane from anaerobic digestion. Furthermore, the ADMEC with applied voltage actually led to greater methane generation than AD only. Furthermore, with the electrocoagulation, we saw a massive removal of the total suspended solids, the total solids, and color from the solution. Also, we saw a significant reduction in the biochemical oxygen demand and the chemical oxygen demand. If you'd like to know more, you're gonna have to come check it out. Thank you.
All right. Hi, everyone. My name is Yusoshi Chatterjee, and today I'll be talking to you about analyzing the effect of poultry litter-derived biochar on ornamental plants. So biochar is a very carbon-rich, nutrient-rich substance that you get as a product of anaerobic pyrolysis. So this is one of the main components, the others being bio-oil and syngas. So to look at this main product of this process, we want to characterize and see what kind of applications can be done with this biochar in both a greenhouse setting and even in field studies. So to conduct this experiment and assess the characteristics, different ornamental bedding plants were chosen. Poinsettias and marigolds, since they have a high markup value within the DMV area, and they're pretty generally hardy plants. So for the initial poinsettia experiment, 5 different biochar inclusion rates were tested, ranging from 0 to 20% at 3 different fertilization rates of 400 ppm nitrogen, 17,517 NPK. And actually, from this experiment, we kind of saw that the inclusion of higher levels of biochar actually stunted the growth of the poinsettias, showed a little more chlorosis, just kind of negative effects of straight on biochar. However, with marigolds, we tried various substrates, slightly lower range of biochar inclusion, tested the feedstock variability of each of the poultry litter that goes into making the batches, and tried nitrogen specific fertilizers. And really that was the key in making sure that there was a difference between the treatments increasing in biochar going from here to here, where you can clearly see more biochar, a little less growth. But with more biochar, we actually saw the highest yielding treatments in the marigolds, as long as we took care of the NPK imbalance, since there is a nitrogen deficit in our biochar. To find out more, come to my poster. Thank you so much.
All right. Hello, everyone. My name is Elad Shtema. I'm a student of Dr. Mitchell Zuckerman, studying the impacts of invasive vines, English ivy, and porcelain berry on nutrient cycling, nitrogen carbon cycling in Baltimore's forest patches. So urban forest patches are self-sustaining systems within urban land use contexts, which contain regenerative forest vegetation. They are shaped by both environmental factors and human technical and social structures as well. Looking at the image on the top right, this forest patch is surrounded by impervious surfaces and faces mixed management. These are just 2 of the human-induced stressors which make forest patches both degraded and sui generis systems. Simultaneously, forest patches provide irreplaceable ecosystem services to the city, such as nutrient cycling, urban heat island effect mitigation, stormwater management, and the provisioning of green space for residents. Looking at the map, these dark green spots are really the only places where these forest patches lie. The lighter green are parks and alike. So this is quite sparse. Baltimore Green Space down here is an organization dedicated to preserving, protecting, restoring forest patches across Baltimore. They have identified invasive vines as one of the greatest stressors to these patches, which are poorly understood and lack adequate management protocols. And they also identify English ivy and porcelain berry as 2 of the most prevalent of these vines. Invasive vines are known to grow rapidly due to lack of natural controls, climbing, choking, and smothering invasive native species, as you see in these images again. They're also generally known to alter litter content, accelerate nitrogen cycling, and interrupt decomposition. However, individual species can be different in their impacts in different regions. It has been recognized in the literature that there needs to be more species-specific studies of plant invasions. In this study, I will determine how these 2 specific vines each impact forests through their effects on trees, soil physical, chemical, and biological aspects, and finally on the overall carbon and nitrogen cycling. The vines will be studied in gradients of cover to inform management of the levels of invasion which necessitate control. because complete eradication is at this moment unattainable. You can find out more on my poster. Thank you.
Hi, I'm Jocelyn Wardrop. I'm a second-year PhD student studying the distribution, formation, mineralogy of manganese redoxamorphic features in soils. My advisor is pedology extraordinaire, Dr. Ravenhorst. So my research focuses on the formation of these features. You may have seen the redox ladder diagram in the past in soil classes, or if you have not, I'm going to briefly describe it to you. When a soil becomes anaerobic, soil microorganisms in the presence of carbon will reduce elements in the sequential order of nitrogen, manganese, iron, sulfur, and then carbon. The reduction in manganese represents a moderately reducing conditions, whereas iron, which is usually used for water table identification, represents more strongly reducing conditions. The reduction and then oxidation of manganese within soils creates these distinct redoxamorphic features that occur at differing portions of the landscape and can occur in the presence or even the absence of an actual water table. My doctoral research focuses on the formation of these features because we do not know the mechanisms with especially the coatings and masses of how these features form. The distribution of these features, manganese features, are not found ubiquitously across all soils. So where are they situated geographically and where are they situated within the pedon themselves across different feature types? Additionally, my research will focus on looking at the mineralogy, which is poorly documented in the literature thus far, and the interpretation. How should soil scientists moving forward focus on identifying, describing, and quantifying manganese redoxamorphic features in soils? So to find out more about these strikingly beautiful black redoxamorphic features, come to my poster.
Are your appetites wet? So thank you for attending here. We're now prepared to recess out into the atrium to examine the posters.